Hello, everybody. Today I'll be presenting on a 2023 study titled A High-Quality Genome Compendium of the Human Gut Microbiome of Inner Mongolians. This study, published in Nature Microbiology, provides really one of the more detailed catalogs to date of the human gut microbiome, specifically using samples from Inner Mongolian individuals. And my name is Abu-Rumman as well. So let's start with the bottom line up front. First, the gut microbiome plays a critical role in the health and pathogenesis of many diseases. That means that the microbes living in our gut aren't really just passive passengers per se. They're more actively influencing everything from digestion to inflammation and overall health. Secondly, much of the human gut microbiome remains unexplored due to certain limitations, but really therein lies the possible new therapies and opportunities to improve precision medicine. Despite major advances in microbiome research, many microbial species in our gut are still unsequenced or poorly understood today, especially in populations not widely understood. And finally, the question driving this study was, can we use a hybrid approach to generate more complete and accurate microbial genomes, particularly in an underrepresented human population such as Inner Mongolians? And really, to answer that, the authors combined long and short read sequencing technology. Short reads are great for accuracy, while long reads capture structural continuity. Together, they allowed the researchers to assemble a high quality, near complete microbial genomes that give us a much clearer view of the gut ecosystem. Now let's give a little brief background, and this is a lot of my sources cited in a little almost literature review for you guys to understand a little better. And the first thing I put was, well, it remains extremely complex, really, to define what a healthy microbiome in your gut looks like. It's really well established that the gut microbiome is essential to human health. And this means that even though scientists still can't agree on exactly what a normal or ideal microbiome looks like, we do know that it plays a major role in maintaining our well-being. And secondly, most microbes, many of them living in our gut, really can't be cultured. And that's the main technical limitation that guides this study. Thirdly, because of this, we need MAGs, or metagenome assembled genomes, to access their genomes. However, of the MAGs that exist right now, prior to this study at least, many are fragmented. So instead of culturing microbes, scientists now extract DNA directly from stool samples and piece together the microbial genomes using computational tools, which we would call MAGs. Now let's talk about why this research matters. Firstly, there is rapidly emerging evidence that links the gut microbiome to health and disease overall, from obesity to inflammatory bowel disease, and even neurodegenerative conditions like Alzheimer's, gut conditions, and others. If we don't improve our ability to sequence, assemble, and link microbial genomes functionally, we really risk overlooking possible pathways or organisms that can influence our understanding of disease risk and overall health. So, in other words, really, if we can't get accurate, complete genome data from the gut, we may be missing microbial genes or species that are actively shaping things from immune function to metabolism or even how we respond to medications. But really, right now, I noted that we're facing two major limitations prior to this study. First is incomplete and fragmented data from short read sequencing. And this technology gives us tiny little snippets of DNA, making it really difficult to reconstruct entire genomes in these little tiny fragments, especially for microbes with repetitive or sometimes complex regions. And secondly, which this study also addresses, is a lack of non-Western population studies. Most gut microbiome data comes from Europe or North American populations, which limits how much we can really generalize about the data we see. There's an urgent need to study these understudied populations, such as the Inner Mongolians, which is a gap that this paper seeks to fill. So now we'll move on to the article and what really that they're trying to set out to answer. And really, in layman's terms, I put that the authors are setting out to answer, is it possible that we can build a more complete picture of the microbes in the human gut through the studying of populations not well represented in past studies, and also by using a hybrid sequencing approach, which includes short and long read sequencing combined together? Now, the methods and the study design and sample collection, to begin, I kind of made it a little bit simpler for us to understand. There's 160 healthy volunteers from Inner Mongolia split into probiotic, containing a probio M8 strain, versus regular yogurt groups. Now, I don't want you guys to focus too much on the yogurt interventions because really these were almost to try and just get their gut microbiome to be active and for better sequencing results. But do note that one of the groups contained a probiotic intervention, which we'll talk about later, was used to kind of validate their pipeline in a way. And these also included collecting stool samples from days 0, 7, and 28. Now, of those 160 healthy volunteers, in which they split into two groups, 80 and 80. 30 participants from each of the two groups were selected for a total of 60, which were chosen at random for sequencing. Furthermore, to note that these investigators were blinded during this experiment and throughout the procedures. They used hybrid sequencing pathway, which we just talked about, but to be more specific, Oxford Nanopore for long read and Illumina HiSeq for short read. These are both very commonly used in sequencing. Now, they assembled the microbial genomes from stool samples through Metafly and NextPolish, more bioinformatic tools. Now, lastly, binning and functional analysis. The contigs were grouped, and we'll talk about more about what contigs are in just a second, but contigs were grouped into genomes through MetaBat2 and VAMB, which are also bioinformatic tools, functional annotation conducted through GutSmash, another tool for bioinformatics, and the main focus here, which we'll get into more detail in the results, was to recover complete circular microbial genomes, which we'll refer to as CMAGs, identify metabolic gene clusters, which we'll refer to as MGCs, as well as prophages and other more functional parts of their genome. Then compared to UHGG, which is the global reference data set for genomes. Now let's start with the results and a little bit of an overview. As we know, in this study, the researchers worked with 180 fecal samples collected from 60 individuals, 3 points in time, and they used two sequencing platforms, ProMethION for long reads and HiSeq for short reads, as well as Oxford Nanopore for the long reads. Now, long reads generated 3.7 terabases of data, while the HiSeq for short reads added another 20.1 terabases of data. This is a lot of data, really, and combining them gives us the hybrid assembly approach, and through this they were able to stitch together longer genome fragments, achieving an average N50 length of 278,000 base pairs. And really just to explain what N50 is, because we'll use this measure a lot throughout this presentation, N50 is a measure of how complete your assembled genome is, and a higher number means better quality, essentially. Now, to validate this, they reconstructed the genome of a known probiotic bacterium that was present in the probiotic yogurt group stool samples, Bifidobacterium lactis probio M8, using three methods: short sequencing, long sequencing, and the hybrid approach. The hybrid approach gave the most accurate and complete genome compared to short read and long read alone. Really, this validates their experiment and their pipeline analysis. Now, one of the big challenges in microbiome research has been assembling complete microbial genomes directly from stool samples. Most previous methods, especially short read approaches, led to fragmented or incomplete genomes, making it really hard to study microbes in their full context. By using their ultra-deep hybrid sequencing method, this study successfully reconstructed 802 complete microbial genomes, also known as CMAGs, or circularized metagenome assembled genomes. That is a huge technical leap. After removing duplicates and applying a 95% similarity threshold to define species-level groupings, they ended up with 134 unique species-level genomes. And really, the truly exciting part was 131 of those had never been published before, adding novelty to this experiment. And this also really shows that this study uncovered a massive amount of previously unknown microbial diversity. So now let's look at a specific example of a CMAG, Victivalis sp. Before this study, Victivalis sp. was a species that researchers could only really study through highly fragmented genome data. But thanks to the hybrid ultra-deep sequencing approach used here, this study was able to assemble a fully circularized genome of Victivalis sp. That means that they recovered the entire genome in one contiguous sequence. And even better, they captured key genomic regions that are often difficult to assemble, such as ribosomal RNA genes, insertion sequences, prophages, and many more parts, which we'll see in the next few slides actually, where I reference this. Now this is figure 1a. This figure shows a maximum likelihood phylogenetic tree of the 134 unique species level CMAGs assembled in the study that I just referenced. Each branch represents a different bacterial species that was reconstructed. The different colors and symbols around the circle provide extra context. They show the phylum level taxonomy, which is up here in the top left, whether a species has cultured isolates or not cultured isolates also noted here. And that's kind of going to show us how novel this experiment is. The black bars extending from the outer edges as well, and the numbers next to them represent the number of individual genomes that they assembled for that given species. What this figure shows us overall is not just diversity but novelty. It reinforces how effective that this approach was at capturing a wide and deep snapshot of the gut microbiome that includes both known and previously uncharacterized bacteria. Now these two figures are figure 1b and 1c. So we'll see in both of these, the outermost circle is just a scale in base pairs, you could kind of say. So on the left here is Victivalis sp, which I had referenced earlier. This is 5 megabase pairs. And on the right Bacteroides A. plebis A, 3.5 megabases around megabase pairs. And this is a large number for their genome. The second ring on the inside, this kind of yellow one, shows the genome of the CMAG assembled in this study. So it's obviously complete, it goes all the way around. And the third ring shows the most closely related genome previously available in public databases. Now, really what's important about this is the blue regions, which indicate where there's greater than 90% identity between the newly assembled CMAG and its reference counterpart, meaning this is what we almost, kind of the blue is what you can consider what we almost know already about this genome, and the white is the spaces filled in by the new data. Now, what you can see here as well, and what's really striking about this map, is it reveals beyond alignment. The colored marks that we'll see here within the CMAGs identify important genomic elements. The green marks are ribosomal RNA operons, red are prophages, and black are insertion sequences. And what we see here is that this study didn't just assemble complete genomes, but it captured complex genome features that are often fragmented or completely missing. And you can see that through kind of the alignment of a lot of these operational units aligning with white, otherwise known as they were not captured in previous data set sequencing. And this gives us really a more holistic and functional, informative, I guess you could say, view of these gut microbes, especially for undercharacterized species like Victivalis sp and Bacteroides A. plebis A. Now, moving on, this slide compares two major data sets. The UHGG, which is the global data set that I've referenced a few times, it's actually called the Unified Human Gastrointestinal Genome Collection, and the IMGG, which is the Inner Mongolian data set that these researchers created using the hybrid sequencing strategy. Now, the UHGG includes over 100,000 gut microbial genomes from people around the world, but many of these genomes are still fragmented. In contrast, the IMGG dataset was produced using both long and short read sequencing, which allowed the researchers to create more accurate, complete, and continuous genomes. They really didn't just improve the existing data, but they also enhanced the reference genomes for 288 species, and even discovered 16 entirely new species from the Clostridalis group, mostly. The IMGG genome were clearly higher quality, and they showed that by two metrics. First, they had fewer contigs. A contig, and as I referenced earlier, and I'll explain it now, a contig is a continuous piece of DNA assembled from overlapping sequencing reads. So fewer contigs means that the genome is less fragmented. More of it has been assembled into a larger, meaningful chunks, I guess you could say. Now, secondly, they had a higher N50. N50 is a statistic that tells us about the lengths of these contigs. Specifically, it's the length at which half of the total genome is contained in contigs of that size or longer. So a higher N50 means that the genome was assembled into longer and more complete stretches, which is exactly what you want to see in a high-quality genome assembly. Now that we've seen how the IMGG dataset improves overall genome quality, let's talk about why that matters more specifically, starting with ribosomal RNA genes, or rRNAs. Ribosomal RNA genes, especially the 16S, 23S, and 5S, are critical tools in microbiome science as they are highly conserved across bacteria, making them perfect for identifying species and estimating abundance. But here's the catch. These genes are often missing or underrepresented in short read-based genome assemblies. But in this study, the IMG genomes did exceptionally well in recovering these rRNA genes. Compared to UHGG genomes, IMGG genomes recovered far more copies per genome, meaning their assemblies are not only longer and more continuous, but functionally richer. What's even more striking is that 85% of these species they assembled weren't even represented in the RRNDB, which is currently the largest ribosomal RNA gene database. Now, this sets the stage for exploring even more functional elements like metabolic gene clusters, which we'll get into in the next few slides. Now here, we're looking at figure 3a, which shows one of the most clear demonstrations of how the IMGG dataset outperforms the UHGG global dataset currently available. Each box plot here shows the number of three different types of ribosomal RNA genes, 16S, 23S, and 5S rRNA, per genome across two datasets. IMGG is shown in green, while the UHGG is shown in orange. What we see here is that the IMGG genomes recovered significantly more copies of all three rRNA genes. This was statistically highly significant, with p-values less than 2 times 10 to the negative 16th for all comparisons. Obviously, a very small p-value indicates that this is not just by random chance. Rather, this tells us that the hybrid sequencing approach used in the IMGG dataset, combining long reads and short reads, leads to a much more complete and biologically meaningful genomes, especially when it comes to functionally important genes like the rRNAs. Now let's look at figures 3b and 3c, which focus on metabolic gene clusters, or MGCs, as I had referenced earlier. These are groups of genes in a microbe's genome that work together to produce important metabolites, things like vitamins, short-chain fatty acids, and amino acids, and many more. So they're really key to how the gut microbes influence human health and interact with each other. Now, on the top left in Figure 3b, we're comparing the completeness of MGCs between IMGG and UHCG data sets. The top panel shows that nearly 100% of the MGCs in the IMGG dataset by these researchers are complete, while more than half of those in the UHCG global dataset are incomplete. The bottom panel confirms this, roughly ratio-wise, at least you could say. IMGG genomes contain about 6 complete MGCs per genome, compared to only 3 in UHCG. So obviously you can see here 100% to a little less than 50%, 2 to 1 ratio, as well as 6 to 3, 2 to 1 ratio. So the IMGG dataset effectively doubles our ability to detect full metabolic gene clusters. Now moving on to the right, figure C, we see a larger percentage of each genome in IMGG is made up of MGC coding regions, which are essentially groups of co-located genes that work together to produce specific metabolites like vitamins, amino acids, or short-chain fatty acids. This tells us not only that the MGCs in IMGG are more complete, as we discovered to the left, but they also make up a bigger share of each microbe's genome, which previously was unknown, obviously, with the global data gene set. Now, shifting to metabolic diversity uncovered by the study. To evaluate the diversity of the human gut microbiome, researchers specifically focused on genomes with high quality and continuity, meaning fewer than 30 contigs per genome, essentially, that they had determined for this specific analysis, which ensures that the MGCs they found aren't just fragmented, but rather they're full sequences with biological meaning. Altogether, they analyzed over 15,000 genomes, and from this, they identified a whopping 97,428 metabolic gene clusters, and more importantly, more than 78,000 of these were complete. That's significant because complete MGCs are much more useful when trying to predict what metabolites a microbe can actually produce. But what's even more striking here is that 48% of these complete MGCs had no match to known gene clusters, which means that nearly half of the metabolic potential they found was almost completely novel. So really, this data set doesn't just improve our understanding of known microbes, it opens the door to new microbial functions that might play roles in digestion, inflammation, and other processes. So now that we've established how many new and complete MGCs this study uncovered, let's take a look at how these metabolic gene clusters are distributed across different bacterial phyla in the gut. This is figure 4a, and it breaks down the types of MGCs present in each major phylum. The clusters were categorized using a tool called GutSmash, which I had referenced in the methods, which groups them by their predicted metabolic products. Now you can see different colors here representing different MGC classes. For example, blue for short-chain fatty acids, red for aromatic compounds, and yellow for putative, which refers to basically gene clusters that are likely deemed as functional, but not yet clearly defined. What's important here is the variation across phyla that they have uncovered. For instance, Firmicutes A and Bacteroidota, seen, I believe, here, Bacteroidota and Firmicutes A, the top two. They both have large proportions of short-chain fatty acid-related clusters, which makes sense because they are known to have roles in fiber fermentation and gut health. Meanwhile, Proteobacteria and Actinobacteriota show more diversity across functional classes, including some more specialized clusters like those involved in aromatics metabolism or nitrogen fixation. A lot of this kind of is already known in I guess you could say, individually of these species, but really it tells us that metabolic potential isn't evenly spread across the microbiome. It varies widely by phylum, and some groups may carry unique pathways that we'd never detect without a complete genome. So now this is figure 4b, which is a little bit more complex, but I'm going to try and break it down so you guys can understand a little better. So at the top, those gray bars show the total number of complete MGCs found in each phylum. For example, Firmicutes A and Bacteroidota had the highest counts overall, seen right here. Now the bottom heat map is where it kind of gets interesting. Each row, each row is a phylum, and each column is a specific MGC type. The color intensity shows how enriched that MGC type is in that phylum. Darker orange and red blocks mean stronger enrichment, while white means not significant. Now, asterisks mark the MGCs that were significantly enriched based on p-value less than 0.05. So really what we see in this diverse picture is some phyla are heavily enriched in different pathways like short-chain fatty acid pathways, while others are enriched in more niche pathways like ethanolamine utilization, vitamin biosynthesis, or even aromatic compound degradation. This further tells us that different microbial groups have specialized roles in gut metabolism, and that certain functions may be restricted to specific taxa. With further studies, we can confirm this. But without high quality genome data, really, we would miss a lot of these associations entirely. Now, shifting from bacteria to viruses, specifically prophages, we'll talk about, which are viruses that integrate into bacterial genomes and really can influence their function, metabolism, and even resistance. From the IMGG dataset, the researchers mined over 21,000 previously unknown prophage genomes embedded in the microbial DNA. That's a huge number, really. To kind of make sense of this, I guess, they use the MIU-VIG standards to cluster these sequences into 13,437 species level viral operational taxonomic units, which we'll refer to as VOTUs. Now, the standards are kind of just standardized. They're just referring to the MIU VIG standards as that's mostly what's used by scientists who are looking to study this area of the genome. And this was based on 95% nucleotide identity across at least 85% of the sequence. Really, this ensures that these are meaningful species level groupings. The quality of these assemblies was strong too, with an N50 of nearly 40,000 base pairs, meaning that these were extremely long, continuous stretches of DNA, much better than what earlier short read sequencing could have achieved. But this is significant because these VOTUs represent entirely new viral diversity in the human gut, which obviously more studies could review some really significant and interesting findings. Now let's take a closer look at what these kind of viruses newly discovered and let's basically take a look at what kinds of viruses these newly discovered VOTUs actually are. Now, figure 5a on the left shows how the 13,437 newly identified viral species called VOTUs, which we just referenced, and of this, only 4.5% on the left here, species level, 4.5% matched to known species, and just under half, 46.7%, could be assigned to known viral families. That means most of these viruses, over 95%, are completely novel or unclassified. Now, figure 5b breaks down the family level assignments. Among the classified VOTUs, Syphoviridae dominates with 73%, followed by Myoviridae at 24%, and smaller contributions from Podoviridae, Microviridae, and Crassphage, and others, each either 1% or below. But together, these charts really highlight how little we still know about the gut virome and just how much hidden viral diversity this study has actually uncovered. So now moving on, let's explore which bacteria are actually hosting these prophages, which we just mentioned. And this is figure 5c. So each row represents a different viral family, like Syphoviridae, Myroviridae, and so on. The colored segments within each row show what bacterial phyla those phages tend to infect. Now here's what's interesting. Both Syphoviridae and Myoviridae, which made up a majority of classified prophages, had a broad host range. You can see this by diverse colors, meaning they infect many different phyla, as you can see. But looking at Crassphage on the bottom, it's basically all pink, as well as Papillomaviridae. It's basically all pink, as I just said, and meaning that's highly specific to Bacteroidota. So some phages obviously have a very narrow host range, while others are generalist, as we can see in this data. Also notably, Firmicutes A, which was the most common bacterial host, especially for dominant phage families. And this is really consistent with earlier slides, where I showed that Firmicutes A is a major gut microbe in Inner Mongolians. Now here's the last figure I'll present for the results, and it's 5e, where we're looking at metabolic potential of prophages, specifically what kind of metabolic genes they carry. Now on the left, we see overall breakdown of metabolic categories. We see amino acid metabolism, which was the most common, making up 2.51% of functional prophage genes, followed by cofactors and vitamins at 1.81%, and carbohydrate metabolism at 1.55%. Now the right side is a heat map showing which viral families carried these metabolic genes. The darker the box, the more genes from that category that family had. So you'll notice a few things. Crassphage stands out for its involvement in vitamin metabolism, for example, while Microviridae and Inoviridae seem more involved in energy and amino acid metabolism. But overall, really, this figure suggests that different phage families contribute differently to host metabolism, potentially affecting gut health in unique ways. Now, moving on to the discussion, we'll talk about a few things. Firstly, this study provided a massive expansion of microbial genome resources. And using hybrid ultra deep sequencing, this hybrid approach really, the study tripled the number of complete microbial genomes available. Reference earlier 802 CMAGs that they had um, assembled, and the study actually states that 225 were previously available, including 131 of those were newly reported species across 11 phyla from those 802 CMAGs. And that shows really dramatically how they improved the microbial taxonomic resolution that we had before. They also enhanced functional insight through the complete gene recovery. Now, the recovery of 78,675 complete metabolic gene clusters, MGCs, nearly half of which were novel, along with the accurate assembly of ribosomal RNA operons, addresses long-standing gaps in existing data sets and enables more precise functional characterization of the gut microbes going forward. Lastly, they revealed uncharted viral diversity in the gut. Really did, and through this they discovered over 12,800 novel viral species, VOTUs that we had just discussed, and 95.5% of which had no species level match. And it highlights the untapped viral component of the microbiome and its potential role in shaping host metabolism, immunity, and disease risk. Now for the concluding remarks, which I'll kind of just read off. Hybrid ultra deep metagenomic sequencing is far more accurate and provides much more complete results for genome assembly. While there's much still to be discovered in understudied populations, clearly there was a large amount of novelty found within the gut microbiome of Inner Mongolian individuals. Lastly, going forward, future studies can build on this novel approach to investigate potential microbiome-associated therapies. Now, these are the questions for our discussion. How might novel MGC, metabolic gene cluster, discoveries influence future therapeutic approaches targeting the gut microbiome? Next, what are some potential ethical considerations when targeting understudied populations, such as the Inner Mongolians, which really is more of a generalized question? What does this new data suggest about the reliability of commonly used reference databases, such as the UHGG? And for example, I included the statistic that 85% of the species in the study were not represented in the RRNDB from referencing the ribosomal RNA genes. How might improved ability to recover CMAGs affect our understanding of microbes as a whole? And remember, CMAGs are circularized metagenome assembled genomes. Lastly, how could we study antibiotic resistance gene transfer in the gut with regards to the prophages detected in this study? Now, here are the sources which I cited earlier, and thank you very much for reading and listening to this presentation.